So we talked about ZAP70, and once it is activated, ZAP70 can have several different effects, which we'll talk about in a bit more detail. But this is a good overview of those effects. So we see here that ZAP70 can then have kinase activity and phosphorylate other downstream proteins. Some examples are activation of proteins called AKT, which leads to increased cellular metabolic activity. It can also activate PLC gamma, which we'll talk about, and this leads to the activation of different transcription factors. And there are a couple other examples we're not going to talk about in detail, but if you guys want to look at how these are related, you can check this out in the book. But essentially what's important is to realize that ZAP70, this one adapter protein, can lead to many different effects, from changes in cell metabolism to cytoskeletal reorganization. And all of these effects are important for T cell activation. So I mentioned PLC gamma. So this is a protein which, when ZAP70 phosphorylates a protein called LAT, it's then able to associate with a couple other proteins. And you can see the beginning of this complex here. And this helps attract the lipids PIP3, which then can accumulate at the plasma membrane. So the different coming together of these different proteins and lipids then allows for the recruitment of PLC gamma. And then PLC gamma is activated by phosphorylation by another protein, ITK. So these are a lot of different proteins. You don't have to remember all the specific names, but just remember that ZAP70 is causing the coming together of a few different proteins, which then attracts another enzyme, which is activated and will cause further downstream effects. So how does PLC gamma work? So it generates what are called second messengers. And these are small molecules downstream, they might be ions as we'll talk about, that can propagate throughout the cell and lead to different signals. So PLC gamma can bind PIP3 and it breaks it down into diacylglycerol, or DAG as you'll see, and inositol 1,4,5 trisphosphate, which will be designated as IP3. And these are two very important second messengers. We'll see some examples in the next few slides of how these two second messengers lead to a variety of downstream effects. So already you guys can see here how each step of the signaling pathway can branch out and lead to different downstream effects. And this is how the simple binding of a TCR to peptide MHC leads to so many different changes within the T cell where they can become activated. So this is another branch point. So here we have the diacylglycerol and IP3, and we'll talk about some examples of how they can cause further signaling. So DAG can diffuse through the membrane. It's a lipid, so it can move through the membrane, and it can recruit additional proteins on the intracellular side of the membrane. So IP3 can also then diffuse through the cytoplasm and bind to the IP3 receptor on the endoplasmic reticulum membrane. So this is a really important pathway because this binding on the ER leads to calcium influx into the ER. So the IP3 receptor will then activate this STIM1 protein, which helps open a calcium channel. So then, because there's more calcium inside the ER than there is outside the ER, the opening of this calcium channel leads to a rush of calcium out of the ER into the cytoplasm. So then we see that this calcium influx can bind to and open additional channels on the plasma membrane, which allows calcium to actually enter the cell. And then we still have our diacylglycerol in the plasma membrane, bringing signaling proteins together, which that calcium influx can activate. So one example of how calcium influx leads to downstream signaling is through the transcription factor NFAT. So here, this is actually not a tyrosine kinase. In this case, we're dealing with serine and threonine kinases. So phosphorylation on the serine and threonine residues keep NFAT in the cytoplasm. So when these residues are phosphorylated, NFAT is stuck outside the nucleus. It's not able to enter the nucleus and cause its transcription factor activity. However, when calcium enters through the plasma membrane, like we just talked about, this actually activates a serine and threonine phosphatase. So this leads to dephosphorylation of NFAT. So you can see here, this kind of greenish protein is covered with several phosphate groups. However, after calcium influx, these phosphate groups are removed by that phosphatase calcineurin, seen here in blue, and this leads to NFAT being dephosphorylated, which then allows NFAT to enter the nucleus through a nuclear receptor and cause transcription factor activity. So here, I know this is a complicated process, but you can see how this branch point leads to calcium signaling, which then, through these phosphatase activities, causes transcriptional changes. So NFAT can bind to the DNA and then lead to different transcription of proteins, which are important for lymphocyte activation. So I also mentioned RAS earlier. So this is one of those small GTPases. And here, it's important to link signaling to what's called the MAP kinase cascade. And this MAP kinase cascade is very important for lymphocyte signaling, but it's also seen in many other types of cell signaling that we won't be able to talk about today. So here, RAS is typically inactive, so it's bound to GDP. However, this diacylglycerol molecule that we talked about is in the membrane, and it recruits RAS to the membrane where it is activated. So here we have one of these GEF proteins mediating the switch from GDP to GTP. So this activates RAS. So now that RAS is activated, it can bind and activate additional proteins. So this is where we get the MAP kinase cascade. So here you don't have to know the specific names of the MAP kinases, but these are some examples. So RAF here is phosphorylated by RAS, which activates it. So this cascade, the generic names are easy to remember in some ways because they just build on each other. So RAF would be what is known as a MAP kinase. So this, the MAP K3 actually phosphorylates, so it acts as a kinase against the next downstream protein, which is a MAP kinase, which then acts as a kinase on MAP kinase. So it's not a very creative naming scheme, but we have this MAP kinase originally, and then you can think of the name of that. So the MAP kinase is the name of this protein, and it's phosphorylated by another kinase. So that's how you get the adding of the kinases onto the name. So MAP kinase is phosphorylated by a MAP kinase, which is phosphorylated by MAP kinase. So again, you don't have to remember the names, but basically know that this pathway is initiated by RAS, so this small GTPase. So that initiates this MAP kinase cascade, and it allows the MAP kinase to be activated and to enter the nucleus again and have transcriptional activity. So it can activate different transcription factors within the nucleus, causing different gene expression. So we also see a different pathway, still building off of that diacyl signaling module, where protein kinase C activates additional transcription factors. So here we see diacylglycerol on the far left recruiting PKC to the membrane. And here it phosphorylates a different protein, which creates a scaffold. So this is an example if you think back to those scaffolding proteins. So it's bringing together multiple proteins that are important for signaling. So this scaffold recruits TRAF6, which makes a polyubiquitin scaffold on itself, and another protein known as NEMO. So here we can see the linkage of a couple different motifs we talked about earlier. So we have our scaffold protein, and this is leading to ubiquitin activity. So it's marking different proteins for degradation. So this polyubiquitin scaffold then leads to the phosphorylation of IKK beta, which phosphorylates I-kappa B, which is the NF-kappa B inhibitor protein, which you guys talked about NF-kappa B a little earlier in chapter 3. So this is just showing some of the details of that. So NF-kappa B is bound to I-kappa B, and I-kappa B, similar to NFAT, keeps NF-kappa B outside the nucleus. So it's just floating around the cytoplasm in an inactivated form, unable to influence gene expression in the cell. So when we have this downstream signaling cascade, I-kappa B is removed, and this allows NF-kappa B to enter the nucleus and cause transcription. So you can see how the polyubiquitination here marks I-kappa B for degradation. So that's how it releases NF-kappa B to enter the nucleus. So this is just another variation of how these signaling mediators, the second messengers, DAG and IP3, lead to transcriptional changes. So another example is PI3 kinase, and this activates different proteins leading to changes in cellular metabolism. And these are really important for lymphocyte activation because these T cells and B cells as well need to be able to utilize glucose and produce different cellular metabolites in order to become activated. So here, PIP3, that lipid in the membrane, recruits some adapter proteins and activates another protein called AKT. AKT is then, it acts as another kinase, and this phosphorylates another protein, which then releases it from its adapter protein BCL2. So phosphorylated BAD or BAD then binds to another protein just called 14-3-3, which allows BCL2 to promote cell survival. So the binding of BAD to BCL2 actually promotes cell apoptosis. So when these two are dissociated through differential phosphate binding and activation, this allows the cell to persist and survive. So this is really important for T cell activation because you want those activated T cells to be receiving signals telling them to stay alive and act. So this active AKT can also have effects on additional complexes, in this case another GTPase, which allows binding to mTOR. And mTOR is another very important broad pathway that you don't have to know the details of, but know that mTOR is important for changes in cellular metabolism. So you can see here some of those effects. So there's an increase in lipid production, increased protein synthesis. We also have more mRNA and protein translation. So you can see here, again, that branching off from the IP3. So we've seen some different effects through whether it's the calcium influx or binding to GTPases. In this case, you can see how the IP3DAG mediators can cause changes broadly in cellular metabolism. Okay, so we've talked about a lot of different pathways. So this is a nice summary of that. So we have TCR or Bcr signaling, and this leads to changes by tyrosine kinases, and those kinases change adapters and scaffolds, and they lead to phospholipases and lipid kinases. So those are those PIP3, which leads to diacylglycerol and IP3. And this causes changes in those downstream mediators, which could be GTPases, additional kinases, or phosphatases. And then finally, this whole long cascade, this leads to changes to transcription factors, cytoskeletal components, or cellular metabolism. So we have all these different proteins which act on each other, which is why this is called a signaling cascade, because each step leads to many additional steps. And together, these can lead to specific changes that are important for T cell activation. And as I mentioned, Bcr signaling is similar, but the protein names are different. The idea is still the same though, so we're not going to be going through Bcr signaling in detail. Okay, so finally we're going to discuss a few co-stimulatory and inhibitory receptors. So these are receptors that are important for modulating the signaling through the main receptor, which is the TCR or Bcr receptor. So one of these co-receptors is known as CD28. So CD28 is required for naive T cell activation. CD28 binds to two different molecules on the cell surface of an antigen-presenting cell. These are known as B7.1 and B7.2. And in order to engage TCR signaling, the T cell must have binding to the TCR as well as this co-receptor, CD28. And we won't go into the whole downstream pathway of this, but CD28 promotes generation of PIP3 again. So since this is similar to signaling downstream of the TCR, this CD28 binding helps reinforce that TCR signaling. So this leads to the maximum activation of the T cell, and it makes sure that that T cell can become activated and respond. So, as I said, the maximum PLC gamma activation here requires both TCR signaling and CD8 signaling. And then it follows through the same pathway that we've talked about with the IP3 or calcium influx, following down to changes in different transcription factors, which help promote T cell activation. Conversely, there are also inhibitory receptors. So, I'm going to start this slide again. So I messed up. Okay. So, on B cells, we have CD40 binding to CD40 ligand. And this helps augment B cell activation and help provided by the T cells. So here CD40 is found on the B cell and CD40 ligand is on the T cell. So these are part of the TNF receptor superfamily, and binding helps promote activation of NF-kappa B and the PI3 kinase pathway. So CD40 on the B cell is analogous to CD28 on a T cell. But in this case, the co-receptor is activated from T cell help. So the T cell comes, interacts with the B cell, like you guys talked about in previous lectures, and this allows binding of CD40 to CD40 ligand, and therefore enhances the downstream signaling. Conversely, there are also inhibitory receptors, which help downregulate and control the response. So some examples of this that you might have heard of are CTLA4 and PD1. So both of these are important targets for cancer, and that's because they help control T cell signaling. So CTLA4 binds to the same receptors as CD28, in this case B7.1 and B7.2. However, CTLA4 actually has a higher affinity than CD28, so it can outcompete the binding of CD28 and take up all the binding of the B7.1 and .2. And this helps block the co-receptor signaling from CD28 and helps control T cell activation. So CTLA4 actually is mostly regulated by expression of the cell surface on T cells. So after the TCR is first engaged, so after you have activation of the T cell through the TCR binding to peptide MHC as well as CD28 binding, this creates an upregulation of CTLA4. So now that the T cell is activated, you actually have more expression of CTLA4 on the cell surface. So this upregulation then starts to compete with CD28 and helps reduce signaling. So this stops the T cell from just having more and more signaling as it binds to its cell surface receptors. So as I mentioned, CTLA4 is an important target for cancer. And that's because in cancer, we often see the immune system stop responding to tumor cells. So this inhibition by CTLA4 and PD1 actually are stopping the T cells from attacking and clearing the cancer cells. So if you give a monoclonal antibody to block CTLA4 or PD1, this then promotes T cell activation, and the T cells of the immune system will then go on to attack cancer cells more. So it blocks this inhibition. And besides T cells, these inhibitory receptors are found across many different cell types. These are some examples here through B cells, T cells, and NK cells. And all of these work through these ITIM motifs. So we talked about ITAM motifs, which are activating, but ITIM motifs are inhibitory. And so when phosphorylated, these ITIMs actually recruit phosphatase of kinases. So then these phosphatases come and start taking off phosphate groups and deactivating proteins in the signaling cascade. So that's how you get the inhibitory effect. Okay, so we went over a lot of different pathways today, but I want you guys to focus on a few themes. So overall, in summary, we saw that signaling turns an extracellular signal into intracellular events. It also involves these enzymes called kinases and phosphatases, which add or subtract phosphate groups, as well as scaffolds and adapter proteins, which help bring different signaling proteins together, and GTPases, which help act as molecular switches in signaling. We also saw that the TCR and Bcr, which are made up of variable extracellular domains, associate with invariant intracellular chains. And this TCR and Bcr signaling leads to transcription factor activation.